with that, we have several minutes left for me to introduce Professor Burkhard Becher, who perhaps is now joining us, as you can see. So Professor Becher is currently, he's a professor and chair at the Institute of Experimental Immunology in Zurich. Early on, he completed his master's and undergraduate degree at University of Cologne before moving actually to Canada to McGill University for his graduate studies, and following that went to Dartmouth Medical School for his postdoctoral work. Professor Becher has contributed an immense amount to the field of immune tolerance, to neuroinflammation, and to understanding cytokine networks and that contribute to both cancer and to autoimmune diseases. We're very excited to have you with us today. And I should also mention that in addition to being an outstanding scientist and mentor, Professor Becher is also a master pizza maker, which I think is something to appreciate in its own accord. So pleasure to have you with us. Thank you for your time, and very excited to learn about more of your work.
Thank you so much, Bayam and Eleanor, for having me. I'm super excited to be here. Thank you so much for the introduction, and to point out what is really close to my heart is, in fact, cooking, because I think in the end, when we are no longer there, maybe people will remember some of our cooking rather than our science, or at least there's some hope to have some lasting effect. Let me just see whether I can put this, my presentation, continue. So I should be able to put my presentation on the screen. I hope you see it now.
Yes.
And I hope you see the presentation mode and not the other mode.
It's the presentation mode.
Excellent. Okay, so I have decided to change the subject of my talk today a little bit. And I would like to talk about two projects which are not in the public domain, and because they are, so that the work is not yet published, two projects which are not yet published. And trust me, that's not for lack of trying. It just happened that it hasn't yet worked out yet, but I think these are very exciting projects, and I'd like to share them with you already right now. And it has to do with cytokines that are very close and dear to my heart, and I'll tell you why in a second. It becomes evident because of my time at McGill University. And the molecules I would like to talk about are IL-12 and IL-23, and you see them here. It's just a review article that I basically cut and paste here, probably somewhat sloppily. But basically what you can see is that they are very similar, namely that interleukin-23 has two subdomains, one P19, one P40, and IL-12 shares this P40 subdomain, but has a unique P35 domain. IL-12 was discovered much before IL-23. And so the receptors are equally heterodimeric with a shared subunit called the beta-1 subunit, and the unique subunit, one to IL-12, and the other one to IL-23. And the biological function is vastly different, but they were confused over the years. And I'll go a little bit more into this. So this is starting with the time when I actually did my PhD, 95 to 98, actually a little longer. But during this time, what happened is it was relatively clear that this molecule, interleukin-12, was a super important pro-inflammatory mediator. And me, interested in tissue inflammation, I followed this very closely. The knockout mice were made by Maurice Gately at Roche, and then lots of papers, lots of people studied this and found that this subunit, interleukin-12 P40, is really vital for the development of all kinds of chronic inflammatory diseases. And I will not introduce experimental autoimmune encephalomyelitis in detail here, but that is an animal model for multiple sclerosis, the most common neuroinflammatory disease in young adults. And EAE models some aspect of that, but then also folks looked at models for arthritis and uveitis and myocarditis. And it was relatively clear this molecule, this cytokine IL-12, is really important. And this was also about the time, as you may remember, we already knew about Th1 and Th2 cells. So this basically was one of these master regulators of Th1 immunity. Here, once again, this molecule. And when I started my postdoc, as a PhD student, I worked primarily on microglia and in vitro system human cells. But as a postdoc, I really wanted to learn about animal models, preclinical models of disease, because I got frustrated with working in vitro all the time, and I really wanted to do something where I can look at a more complex system. And so going there, I ordered mice from Jackson, namely the IL-12 P40 knockout mice, and tried to study the role of brain resident cells in controlling immunity through IL-12 in inflammation. It turns out Jackson didn't have the P40 knockout mice. Instead, they sent me the P35 knockout mice, and I said it doesn't matter because it's the same molecule. But it turns out if you look at this here, the P35 knockout mice were constantly getting super sick, and they don't have IL-12, but they get very strong EAE, and not just that they're not resistant, which is what I expected and what you can see was expected from the P40 knockout. These P35, the specific IL-12 knockout mice, got very severe EAE, and many of the mice died. And so just, I'd like to recap this a little bit. So IL-12 was discovered already in 1989, and IL-23 was discovered only after I've started working on IL-12, not knowing that it was actually IL-23 I was working on. Both molecules, IL-12 and IL-23, are thought to be highly pro-inflammatory, and there's clear evidence that both are what you would consider a canonical pro-inflammatory mediators. What has crystallized over the year, IL-12 is great in driving anti-tumor immunity, whereas IL-23 seems to be vital for autoimmunity. And now it gets weird, because IL-12, as I just showed you, is super immunoregulatory in autoimmunity. And as I'll show you later on in my presentation, IL-23, on the other hand, has pro-tumorigenic properties. So this is where it gets weird. You have a molecule, so they have much in common, but they regulate autoimmunity and anti-cancer immunity completely the opposite way, which is very bizarre and has until today not been resolved. So that's what I'm going to talk about today, namely primarily this finding here, where, as I mentioned already earlier, mice that lack IL-12 are hypersusceptible to develop this autoimmune brain inflammation. And mice, as you can see here, that lack IL-23, or either these two subunits, this is the specific subunit, they didn't develop this papilloma, these papillomas in an induced model here. And this was already published also in 2006, so quite a while back, almost, not quite 20 years, but like 15, 16 years ago. And we have until today no idea why this is. Why is IL-23 promoting tumors, or why is IL-12 preventing and regulating brain inflammation? So let's start with the brain inflammatory part, and I'll share here this is Mirto, is a postdoc in the lab, and Sarah was a postdoc. She's now a group leader at the Institute, and our collaborators in Berlin, Frank Heppner and his team. Together we did this experiment. We basically have to make see whether I can make you all disappear from the screen for a second. There you go, I have more space now. So we made a conditional IL-12 receptor beta 2 allele. In other words, we wanted to find out which cells need to sense IL-12 in brain inflammation. And so this is a knockout first allele that allows us to specifically delete the IL-12 receptor across different cells using inducible Cree or using conditional Cree drivers. But at the same time, this mouse can actually serve also as a reporter for the IL-12 receptor beta 2. So all IL-12 sensing cells should light up in these mice as well. And so the first experiment that we need to do is to find out whether our mice actually work. And I think this is a very, very boring slide. I'm showing you we made the IL-12 receptor conditional and it worked. The latter, however, you should know is not something that happens. It's not automatic. It's not an automatism. It happens often that we make quite a few mice that do not work as expected or as desired. So here, we basically deleted the IL-12 receptor from every cell, made like essentially the same as a full knockout mouse. And as you can see here, when splenocytes, I think, were stimulated with IL-12, they did not induce phosphostat-4, which is downstream of the IL-12 receptor, and that's basically the same data here. And more importantly, and most critically, when we induce, and this is a little trick, we have to induce relatively mild EAE. Our IL-12 receptor beta 2 knockout mice die when we induce regular strength EAE, if you wish. So we reduce the amount of pertussis toxin, which is a little dirty trick that EAE-ologists are using to induce EAE in mice. So we had to reduce it to get a relatively mild EAE in the wild type, but in the knockout, as you can see, they are indeed hypersusceptible and get a much stronger, much more severe EAE. And the numbers here are relatively clear that this is not a fringe phenomenon, but this is really robust. This I showed already, but oh yeah, now maybe I should show this. As you can see here from every review that we've seen so far, every paper on the subject talks about IL-12 sensing in NK cells, so in lymphocyte, NK cells and ILCs, and then of course in T cells. But there's little known about anything else. So the first thing we expected, of course, was the T cell compartment. CD4 T cells are vital for the development of EAE, this autoimmune model here, where you drive it through immunization with the self-peptide and complete foreign adjuvant. And as you can see here quite clearly, when, first of all, we have a rather specific targeting of the CD4, of the T cell compartment using CD4-CRE, so CD4 and CD8 T cells, canonical alpha beta T cells, do not sense IL-12 anymore. However, this has no impact on disease development, as you can see here. The mice develop perfectly normal, perfectly fine EAE, as if we hadn't touched IL-12, which is a bit surprising. To some extent, it's surprising because, you know, we can't really make good so-called Th1 cells here. And nevertheless, in the absence of the Th1 developing alpha beta T cells, we get perfectly healthy EAE. So you don't need IL-12 in the T cell compartment, but also the mice don't get sicker at all. So they don't get higher disease levels. And just to make sure, we kicked it up a notch. We basically decided, what if we delete IL-12 sensing across all hematopoietic cells? So every leukocyte in these mice cannot sense IL-12 anymore, and yet we get perfectly healthy EAE, not more, not less, just like the wild type. And that pretty tells you that hematopoietic cells do not need to sense IL-12 to get perfectly normal EAE, and that hematopoietic cells are not responsible for this IL-12 mediated tissue protection that we see in EAE. And well, it has to be, obviously, I just said that the protective feature of IL-12, so the hypersusceptibility of the knockout mice, has to be explained by a cell type that is not part of the immune compartment. And I have to say I'm pretty badly colorblind. Payam knows this already, or maybe no, he hasn't heard this from me. Because I'm colorblind, I asked the student to write down a few things that I should say about this reporter mouse. So I asked her to do this, and I never really know how to get the feature working for me, that I have the notes. So I basically tell you what she's written here. She said, Burkhard, I know you're colorblind and don't understand histology, but you really have to show this slide and say, IL-12 receptor beta-2 protein within the steady state, specifically in neurons and oligodendrocytes. And we already find this in the healthy brain. And then there are colors here that are referred to as magenta and cyan, and I don't know what that means, but I'm sure you can appreciate it. But she says, and that's important, that the Purkinje cells in the cerebellum showed a very, very strong IL-12 expression. So whatever that means at this point, what I can tell you is that we find strong reactivity within the central nervous system in non-immune cells, namely in neuroectodermal cells, namely neurons and oligodendrocytes. And so just to wrap this up a little bit, so to make sure that we know we're on the right path, we decided what happens if we take IL-12 sensing away from these brain resident neuroectodermal cells. And the answer is we get a phenocopy of the full knockout. Essentially, we can here clearly show that if IL-12 receptor, well, IL-12 sensing is deleted from the brain, the mice get the strong EAE phenotype, namely the one that we get also in the, as you can see here, basically, it's just what I'm talking about. So like the full knockout. So it has nothing to do with immune cells per se. It's actually not really relevant. It's not immunity. We see tissue protection through IL-12 in the brain. And there are some more color pictures for you to show that demyelination is changed as well. And then that's what you expect. Now, the problem that we then had is to say, oh, let's look at the single cell with single cell resolution. Maybe we can find out which of the cells in the brain or which of these neuroectodermal cells and how they what is the impact of IL-12 on the cells, on the transcriptional landscape of these cells. And so we wanted to do obviously a rather deep, detailed single cell analysis of the brain, but it turns out we've done that in the past. And as you can see here, there are no neurons in the brain if you make a single cell suspension. So this is work where we looked at the myeloid compartment. We see microglia, we see all kinds of other cells, the so-called border-associated macrophages in the brain, and this work is long out there. But as you can see here, there aren't a lot of neurons, and that's the reason for this is neurons do not, in adult mice, do not make a nice single cell suspension where you could look at the transcriptome in the single cell level or something. So we decided to go, so as I just said here, that's the bigger challenge we had, is that we don't have the neuroectodermal cells are too fragile. So we had decided to go for single cell nuclear sequencing, nucleus RNA sequencing. And the other problems that we had is in the steady state, we do not see a lot of IL-12 in the adult mice. So we thought, where's the source of IL-12? And so this turns out to be monocytes. During EAE, monocytes will start invading the brain and they make a lot of IL-12. And that's basically the time point, the moment, so long before the mice show any clinical symptoms, we induce EAE, we sacrifice them before they get sick, the moment we know that we have monocytes in the brain, and then do the experiment. And as you can see here, the good news is, yes, we do find a lot of neuronal cells. In fact, they outnumber the microglia compartment. For an immunologist like myself, that was a bit surprising how much stuff is in the brain. But there are all kinds of different neuronal cell populations. There are quite a few other glia cells, including our oligodendrocytes. And as we expected, we do find the IL-12 receptor in the steady state already in these cells, for instance, and particularly strong in these Purkinje cells. And then we did a couple of things to analyze how this could influence brain inflammation. So IL-12 sensing in neurons, for instance, how it could make that change the level of brain inflammation. And so we looked specifically at the producers and targets of the ligands. So we wanted to find out. So this is a niche net analysis, which is just an approximation. It doesn't give you so much, but what came out on top all the time were neuron intrinsic protection programs that we could reveal in the presence of IL-12, and they were lost in the absence of IL-12. And the same is true for trophic factors, so brain trophic factors, factors that protect neuronal cells and oligodendrocytes. And this is basically, I don't want to bore you with the details here. I hope the work is going to be out soon. But basically, it is rather uncanny on how strong the effect is of IL-12 on neuronal cells, and that IL-12 driving neurons really make them look significantly happier and better. And there are a lot of things that are now emerging in this regard. So there is a role for this old-fashioned inflammatory immune cytokine in the function of the central nervous system and in the healthy central nervous system, and in immunoprotection, tissue protection through this cytokine. So basically, this is just a summary here. What we said, we see a lot of interaction between neurons and oligodendrocytes driven through this IL-12 pathway. And we think that neurons are the primary target of IL-12. And to us, this is basically something we find rather exciting, because not just that it's some novel idea about the role and function of IL-12 in inflammation in general, but also it for me very satisfying, because it finally explains why my experiments as a postdoc failed and why these mice get so much more EAE. Having said that, I'd like to, as I promised, I'll move on quickly, and this is also relatively short. This is the other coin, the other side of the coin, namely the role of IL-23, the sister molecule. We know it's involved in multiple diseases, including in particularly strong in psoriasis, where now targeting IL-23 in psoriasis really basically completely blocks psoriasiform lesion formation. It's a very powerful drug, so the neutralization of IL-23. But again, it has very strong tumor promoting properties. I've mentioned the paper earlier here already. This basically was the first paper that shows that there's this rather strong increase in tumor burden, or the loss of tumor growth in the absence of IL-23. But it's also seen IL-23 is very very prominent across many cancer types in human cancer, not just in mouse cancer. So the question here was just the same as before: why is IL-23 promoting tumor growth? And this is the work of Pascal and Tobias. Pascal left the lab already. She's now a postdoc in Israel, and Tobias is a hematologist in my group and never really thought that he would work on this project, but it just happened that it just became fascinating to him as well. And our collaborators are Mo Uka in Seattle, Thomas Korn in Munich, and Sonja Tugas, also group leader at our institute. And so Mohammed made this IL-23 TD tomato mouse. It's a knock-in of the human IL-23 receptor cDNA into the murine IL-23 receptor locus, and this mouse functions fantastically. It's probably the best reporter for IL-23 receptor we've ever seen. And to make a long story short, if we look in tumors, and this is just the B16F10 melanoma just injected into this intradermally, we find all these infiltrating lymphocytes, but there are many, many of them. So we knew gamma delta T cells, and the population of gamma delta T cells would make the IL-23 receptor because they do so the moment they are in the thymus already. But we found a very prominent population of regulatory T cells in the tumors to also make the IL-23 receptor. And if you then compare the IL-23 receptor positive and negative Tregs in the tumor microenvironment, you'll see that in the presence of the IL-23 receptor, you see this very, very strong, let's say, effector Treg program, high levels of GITR, and you know, basically here's the profile. You see this increase in ICOS and PD-1, etc. So they look more like effector T cells than the IL-23 receptor negative. So the transcription and the proteome of these cells is fundamentally different. What we then decided to do is say, well, let's repeat the experiment we had earlier. We had made the IL-23 receptor conditional mutants already, and basically decided, what if we get rid of the IL-23 receptor only in Tregs? What would happen there? And this is the experiment. And this is, again, basically, we get a phenocopy of the full knockout. So all you really need in the mechanism by which IL-23 drives tumor growth is through regulatory T cells. And we see this here in this, let's say, B16 model, which is not very immunogenic. There's another melanoma model called the Yammer 1.7, and we see exactly the same phenotype, even in a more immunogenic model. But also here in this colon cancer model, we see exactly the same thing. So IL-23 appears to mediate its effect exclusively through regulatory T cells. And coming basically now close to the end, we found there is something interesting about Tregs. As you probably all know, the FOXP3 gene is actually located on the X chromosome. So females are, because they suppress the other X chromosome, only one is going to be expressed, are natural mosaics in a stochastic way. We find IL-23 in this particular mouse, we find IL-23 receptor positive and negative Tregs in these females side by side. And luckily, because of this genetic trick, we find that that we can find the loss of IL-23 receptor will also coincide with the YFP label, which is so every IL-23 receptor negative Treg in this mosaic mouse will be YFP positive. And so we can now very easily, by virtue of mosaicism, like induce tumors in these mice and have receptor positive and negative cells side by side, can sort them out. We did some single-cell RNA sequencing. I'm not going to go into this, but what is important here, this is just simple, simple, pretty high dimensional, but still flow cytometry, where we see this very, very, I think this is a rather impressive, to me at least, this program. You see higher levels of ICOS FOXP3, GITR, CTLA4. So the list goes, whenever you think of what makes a Treg a canonical Treg and how does it function, all the molecules that are basically downstream of the FOXP3 are basically dramatically reduced when IL-23 cannot be sensed anymore. And we find pretty much the same thing in human cancers. We find if we look across, this is here in this wonderful resource, these are 30 different cancer types. And whenever we look at IL-23 receptors, we find an even more prominent production of IL-23 receptor in the Treg compartment. And the Tregs that don't make the IL-23 receptor, again, look very similar, as I said earlier. So they look weaker and they look not as, let's say, not as effective as effective Tregs. And there is more data, and hopefully, so actually last night I received the review, and it's the reviews, and they're not horrible. So I think we can possibly answer this, and all the resource will be online. But essentially, what I'm trying to say here is that we know in the tumor and microenvironment, tumor-associated macrophages produce IL-23. So there's an inflammatory stimulus. We think that this attracts Tregs actually, and not saying it acts as a chemokine, but it acts a bit of a survival and differentiation factor. In the absence of these of the IL-23 receptor in Tregs, Tregs don't work so well, and then tumors don't grow so well. So we have a lot of and it actually leads to complete reprogramming of the tumor microenvironment, both by this massive increase in effector T cells in the absence of IL-23, but also we see changes in the myeloid compartment that it's not worth discussing here right now. So with that, basically making sure and being cognizant, I don't want to speak too long, and I so much appreciate that you're listening to me. This is still, unfortunately, an old picture because of COVID. We haven't had a chance to take a new picture in the lab. So most of the people here, maybe with the exception of Pascal and somewhere this is Pascal here, and Myrto is also here, but I just don't see her right now. That's right next to her, that's Myrto. Other than that, nobody is in the lab anymore. So, but those I've talked about. And again, I want to thank you so much for listening to me, and maybe there's some time for questions. Thank you.
Thank you, Professor Becher, for an exceptional talk. I really enjoyed it. Just a note to everybody else who's here, we see our global participants all introducing themselves in the chat. I also encourage you to take this opportunity to ask questions, and we'll moderate these and share them with Professor Becher. I actually have several questions myself, so if that's okay, while the questions trickle in, I'll ask a few. To start with the latter section first, I was curious if this is specifically, the RR gamma T subset of Tregs that might be involved, considering the mucosal kind of nature of some of these tumors.
Yeah, so we don't. That's it's a. I have to say it's an excellent question, something that we didn't consider as much as we should have. We are doing it right now because you're not the only one asking the question. I thought you received the paper, the reviews yesterday. It's one of the things we need to deal with. I don't have the answer yet, but I think I think I have the same suspicion.
And in that sense, I wonder in let's say antibiotics treated mice, how these, because that kind of compartment is very quickly affected by the triple antibiotic cocktail. I'm curious to know. I suppose if there's an effect there, I'm sure there is.
You mean by treating with antibiotics, you said?
Yes. Maybe prior to tumor induction to see if this subset will still infiltrate as much and play as big a role. So then considering if microbiota might play a role in their recruitment. And
that's an interesting, let me write this down. It's actually a really cool question. I don't know the answer to this. It's possible, obviously, because of the link to Rho gamma and the Tregs in the gut. Mechanistically, that's actually something really, really curious. I don't know the answer, but this is something we will follow up now.
There's a paper on that note that just came out last week in Immunity about muscle injury and recruitment of Tregs kind of directed by gut microbiota. So it was quite an interesting link. The first section was also quite fascinating to me personally, and I'm sure to many of our attendees. So I had a few questions on that. And the first being, I was wondering, there's also a lot of implication for alarmins like IL-33 and especially early phase of neurodamage. And I'm wondering what's the interplay between IL-12 and 33 and how they...
I don't know by heart. You're not the first one asking this, and it's on the list. This is actually on the list of our sequencing. IL-33 was rather prominent. I don't recall exactly what it looked like and when this happened, but we saw this in the single nuclear sequencing. The problem is I don't recall that we've seen 33 in the in vitro analysis when we basically just stimulated neurons. Maybe because that's the problem is no matter how you look at, as much as I like the in vivo experiments, the big difficulty that you're having is with in vivo experiments, it's always a mess, which is what you want. You want this complex interaction in so many players, but that is then can potentially mask the role of the direct effect of IL-12, let's say, on these neuronal cells. So we did this in vitro. I don't recall, I don't have the answer for IL-33 right now off the top of my head.
And very last question from me is, in terms of the chemokine receptors that are critical to infiltration of the immune cells, and what are your thoughts in this? Does IL-12 modulate that at all?
So there is a great paper by Doran Merker in Geneva. It was published a few years back, and he uses a significantly different system. But he does find a very strong link between CCL2 and IL-12 signaling, if I recall correctly. So the key here is that CCL2, which is the ligand to the CCR2, and CCR2 is required for mobilization of monocytes into inflammatory lesions. And I cannot stress, that's what I initially want to talk about. I cannot stress enough that we think that these are the primary mediators of immunopathology across various inflammatory conditions are actually not T cells, but in fact the monocytes mediate the pathology. I'm not saying T cells don't drive the system, but the execution of tissue damage, that's in our opinion, and I think the plot's thickening quite well, are actually monocytes.
This is fascinating. Thank you so much. I'll pass on to Eleanor, as I know she also is keen to ask some questions.
Thank you. Great talk. Thoroughly enjoyed it. So I have just a couple of quick, short questions. My first one is, you talk about, IL-12 being neuronal protective, but I wonder how you manage that in the context of demyelination and myelination. What's the actual impact of IL-12? Does IL-12 have any impact on remyelination?
Don't know. We only looked at, that's an excellent point. So maybe just for those of you who are not familiar with this model, so we do see this acute inflammatory response. We see the formation of edema and monocyte mediated damage into the brain, and then we see subsequently to that a loss of myelin. In the models we're using, and being in Switzerland, where we are not allowed to keep the mice any longer, so remyelination does not happen. The mice have to be sacrificed prior to them recovering. So that's just not something, at least in Switzerland, we are not allowed to study this. It's just not possible. There are, however, I'm glad you asked, however, there are models of non-inflammatory demyelination. As you know, you can poison the mice with cuprizone, and then you get demyelination of the corpus callosum. And that is, of course, that's an obvious one. We have to check that. It's a great question. We don't know the answer, but I really want to know what's the role of IL-12 in recovery. And in fact, I actually think it may even be more important after all than it is in the acute disease.
And a quick question about, why don't we see this based on the FOXP3 and the Treg story and IL-23? Do we see, or perhaps I should say, why don't we see the sex bias in cancers?
Say that again. Sorry, there was an interruption.
What your comment about FOXP3 being on the X chromosome and IL-23 being pro-tumorigenic, why don't we see this sex bias in promoting cancers?
Oh, because, okay, I see. Now, so here in this case, we use it's just a trick that we use. It's an artifact of let's say, not really an artifact, but I think if you have this, the X chromosome, I mean, I'm not sure we don't see this as sex bias, but the X chromosome, it basically just blocks the other X chromosome. So I think there is no major difference in FOXP3 expression between men and between the sexes, to my knowledge. So I don't think there is a major difference. However, in this case, we have, of course, the advantage of having this mutation, and therefore we have a mosaic. So if there is no mutation, I don't think you'd see it. If you have a mutation, then you would be able to see this bias. Interestingly, the mice are in terms of the total number of IL-23 receptor positive cells, are relatively normal and healthy, and there's no phenotypic difference that we see in these mice. It's just a trick for us to sort the cells because they're yellow. So that's just a little trick.
Actually, there is a human syndrome, and I'm not sure what it is. I can't recall, where you do see a FOXP3 mutation, and there is a sex bias there in terms of the phenotype. Anyhow, I'm going to get off this so Payam can deal with the other question.
Sure. Thank you, Eleanor. So we do have a question from Muhammad Abdullahi, who is also curious to understanding the function of IL-1 in this context of neuroinflammation.
Yeah, IL-1, okay, so I'm glad you're asking. In the context of this IL-12 effect, it doesn't play an important role here. So IL-12 doesn't have a huge effect on the IL-1 production that we see in the brain in general. I mean, you can always make the argument we have more or less inflammation. More inflammation, more IL-1, less inflammation, less IL-1. So that is probably an effect, but we don't see a direct effect mediated through IL-12 sensing. Nevertheless, IL-1 is absolutely vital for the development of the inflammatory lesion. In the absence of IL-1, we see a dramatic protection. So you do need IL-1 beta. It's really, really important. And it's, again, another molecule delivered by monocytes primarily.
And maybe finally, sorry to ask so many questions, but another thing I was curious about is Tregs are known to express quite high levels of IL-12 receptor, particularly actually human Tregs as well. And knowing that these are also recruited in the context of neuroinflammation, I'm curious if you also
whatsoever. It does not have any impact, at least not in the animal
not
model.
in this EAE model. I see.
I mean, I understand. So that's, I mean, we know that the IL-12 receptor is much more and much better expressed in lymphocytes than in brain resident cells. But we've only recently started to appreciate that as immunologists, we may just be a little bit myopic when it comes to looking at other tissues. I, for one, call every non-immune cell a stroma cell just because I don't know any better. So now I'm learning that not all non-immune cells are stroma cells. Stroma cells have a specific role, and that we need to learn maybe that immunology happens outside of leukocytes as well. So for me, the surprising thing was, and again, I'm not blaming immunologists in general to be myopic. I think maybe I am more than others. I never considered an activity outside of the lymphocyte compartment. I was sure the CD4-CRE and the VAV1 would give us the results. I was 100%, I had everything lined up in my head on how the story would go next. And then you have this TH1, you have this GM-CSF change, and all these things were lined up in my head, and nothing materialized. It was rather frustrating that I was utterly wrong from A to Z. So the brain and some other tissues are just part of the whole gamish of immune communication that we tend to neglect.
Thank you so much, Becher, for a wonderful discussion. This was thoroughly enjoyable, and we really, really appreciate your time and participation in our conference.
Pleasure. Glad that you invited me. I'm very happy to be here today.